Hi, I'm Jeff with Echo, and today we are going to be demonstrating cell counting. This is for our customers whether they have already purchased cell counting or if they're interested in our free 30-day trial. In this video, we will be covering 4 different topics. These include unlocking your software, preparing your samples, the microscope guidelines, as well as the software overview. With your microscope, you should have received a cell counting unlock USB drive, as well as 2 cell counting slides. Other supplies needed for this preparation will be a pipetter, pipette tips, Trypan Blue, and cells. To install cell counting, launch the Echo Pro app. You can see here in the bottom left-hand corner, there is no button saying cell counting. So therefore, we will go ahead and grab our USB drive and plug it into the back. Once plugged in, we will go back to the home screen of the iPad and relaunch the app. You will see now that cell counting has been unlocked in the bottom left-hand corner. To begin sample preparation, go ahead and remove your cell counting slide from the packaging. And then we will mix 10 microliters of our cells with 10 microliters of Trypan Blue. Mix them together and add 10 microliters of the mixed solution to the cell counting chamber. For the microscope, we want to be sure that it is in an inverted configuration with a 10x objective. We want to be sure that the condenser diaphragm is all the way to the right, showing that it's completely open, as well as having bright field on the condenser setting. After preparing your sample, we'll go ahead and place it on the stage and launch the Echo Pro app. In the bottom left-hand corner, you'll see the cell counting button that will open the cell counting software. Once opened, we will go ahead and auto white balance with the sample present to remove the blue background caused by the Trypan Blue. We will close down the illumination aperture to 10% and we'll tap the screen to auto expose. What you're looking for is a nice bright background with nice strong black outlines around each of the cells. After you get that, we'll go ahead and make sure that we're focused correctly. What we're looking for is a nice white center versus a dark center, such as that. So we want to bias it towards the white center, and then we hit capture. After you've captured your image, you can see here all of the live cells are detected in green, and all the dead cells have a red circle around each of them. We can review each of those cells by panning here. If you want to bring up a full gridded menu, we can do that. We could sort by the largest, the smallest. We could select all. We could make various changes right here. Ultimately, if you want to zoom in on a particular cell, we could do that. If you believe this is something such as debris, for example, we could go ahead and remove it and make those changes accordingly. After reviewing your image, when you're happy with the results, we could also gate according to cell size. So we could change our lower gate As we gate cells out, they will show up with an X in them. So you can see all these cells were gated out of the cell population. After we modified our initial cell count, if you want to capture multiple field of views and have them averaged together, we can simply hit the capture button at the top to bring you back. We can change our field of view, and we can go ahead and hit capture. And now you can see here we have our original count as well as our second count. And our live dead total have now been averaged, and we can do this up to 8 different times to get a nice data set. So you can see we have all 3 here. As you continually add more and more images, they will sequentially add below. When you are all done with what you like, we can go ahead and generate a report by clicking our little hidden button up here and hit Generate PDF Report. After it's generated, you can choose how you would like to get the report off the unit through either USB, Dropbox, or even AirDrop. So we could AirDrop it to another Apple user. Or if we hit save to USB, if you have a drive attached, it will go ahead and show that. After all of your data has been collected, simply click the save button to save that data set. We can go back to the main menu by hitting exit in the top left-hand corner, and then can review our cell counting data by opening our image here. And we can see if we hit open cell counting, it will bring you back in with your data to where you can continue editing or review. Thank you for your time today. Please reach out to your local Echo rep if you have any questions on cell counting.